Welcome to today's session where we're focusing on IPM in vegetables grown outdoors. For those of you who don't know me, I'm Rosemary Collier and I'm from the University of Warwick in the UK. So obviously this is an online meeting, and so we sort of have to have a few rules. Sabine, have you got your slide about the
show it?
Yeah.
It takes some time to switch it.
Okay, so I'm sure you're all used to doing it anyway, but please could you, while the presentations are being given, please could you mute your microphone and video. Please put your questions in the chat, and then we'll ask the questions. There may also be opportunities for you to ask them yourself, but at least we know how many questions there are. In terms of the presentations, then please open your PowerPoint in presentation mode and share your screen. And then finally, we'd like to record the presentations. So if you're not happy about that, please can you indicate it in the chat. And it looks like Utu's joined us.
Oh, it worked perfect.
Maybe.
Yeah.
I can't tell.
The people in the lobby, so
yeah, excellent.
Yeah.
Okay, so the way we'll run the session is that we'll have a presentation, then we'll have a question and answer session about that presentation before we move on. We've got quite a few presentations to get through, and we'll just see how much time we've got left at the end in terms of what sort of a discussion we have at the end. I guess the overall aim is just for us to make contact with each other, hear about what's going on, and identify any sort of possible welcome, Uti. Identify any possible areas for further discussion, collaboration, and also we'd like some suggestions about whether this is a useful thing to do, and if it is, then what sort of form it should take in future. Right, so we'll move on to the first presentation, which is by Sabine Pollet from Inagro, and Sabine's going to talk about Smart Protect and Smart IPM in vegetables.
Yes, thank you, Rosemary. So welcome everyone. For those who don't know me, I am Sabine Pollet from the Inagro Research Institute in West Flanders, Belgium, and I am the coordinator from Smart Protect, the EU European thematic network on smart IPM in vegetables. This is a project with 15 partners from 12 countries. It's running for 3 years, and Rosemary is one of our work package leaders. And also welcome to my other partners who are joining this meeting as well. On this map, you can see the partners from the different countries. And a very short introduction on Smart Protect. Actually, it's a thematic network on smart IPM tools. It follows a multiple approach level. And one of the main things we started with was the knowledge collection of all IPM tools. That took almost 1 year for us during quite difficult times during COVID, because there were no fairs or any events to attend. So all the collection we need to gather together by email or phoning companies. And right now we are doing the evaluation of these techniques by doing a benchmarking exercise. And the last year of the program, we will be fully programming a lot of exchange events and webinars, courses, and demonstrating smart tools. And of course, during the whole project, we have the knowledge management. To start with the knowledge collection, this was led by Rosemary. We have gathered quite a big group of techniques, and we decided to form 4 subgroups on that, because we had a lot of techniques which gather in the application subgroup, Then we have decision support. We have diagnostic and detection and monitoring. And here you can see the different subgroups. So for the monitoring techniques, we have one on the insects and one on the crops. Decision support, there's a big there's one group, but for the application techniques, we have the sprayers. We also have sprayer drones, UV systems, or the distribution of beneficials. This was the status on February 8 last week. It's continuously changing, because if we have not enough information to share on the platform, we are deleting the technique, and sometimes we have new techniques was coming up on the platform. We also decided to develop an IPM pyramid for Smart Protect, because we wanted that all partners use the same definition on what they understood for IPM. And you know, as many people of you are working at IPM, that you always start at the bottom of the pyramid, where you start with preventive measurements such as crop rotation or the resistant varieties. And then you come up with decision support. If it does not work, you go higher in the pyramids, and you're doing control measurements, mechanical, physical, or natural. And if this still doesn't work, you start with biological control. And the last top of the pyramid is then the chemical control. And all our tools we are collected, the range of the tools covers the whole pyramids. How did we work on that? We started with an internal database, and each of the partners had a login, and they could edit and enter a certain tool, and we could follow up how many tools we had and who has given the information. If it was not sufficient, we asked them to inform at the company to have more information on certain tools. And we are currently updating the platform so everyone can edit who has editing rules, and then we can add some new pictures, or if we receive new information, or a YouTube video. Now, our Greek partner, Agenso, who is responsible for building our website, they have transferred all these techniques from the database, internal database, to the platform. And if you go to our website here, you can find the link. On top of it, you find the link to the platform. There's also a link to our newsletters. So for people who have interested in smart IPM techniques, please register our newsletter. If you click on the platform, it's freely available. And the good thing on it is that we are currently translating in 12 different languages. So for the moment, everything is in English, and we are starting each week to translate 10 techniques in French, German, Dutch, Portuguese, Spanish, and so on. When you first enter the platform, then choose your language. You have search solutions. So if you are looking for a certain technique, you just type in the word. Or if you want to have more information on technique types, you can click on that button. If you want to have more information on the monitoring or on the application, you can see here the number of techniques. You can also filter by application range. So if you are only interested in fungi or in insects, just go to the insect button, and then you find the techniques who are working on insects. How does it look like? If you have typed in a solution or you found a solution, then you find the title. So here you have an example of the Lumion UVC robot. You have a picture, and then you see the TRL level or the farm scale type. Can you use it in a big farm, or it's only for a small farm? It's for greenhouse of open air. And then you have a short description. You have always a link to the website, because we want to keep the description as short as possible. If you are looking for certain techniques, you don't need to have at the first sight a lot of information. You can see the crops in which it's used, the countries in which it's used, the species, if it's working only on a powdery mildew, or if you have other species as well, and then some technical requirements. If there's a need for a training, sometimes you find the cost details, sometimes not, because this is more customer made. Here there's an example of a cap trap. So you find again the link to the language, you have description, the websites, and then some extra information. It's very user friendly. Also YouTube videos. If you need extra support, yes or no, or the cost of the support as well. So I would invite you to have a look at the platform. If you have any remarks, please let us know. Maybe we can improve as well. If you go to the website, you will also find a button on the vegetable IPM, and there you find some more information. We have some fact sheets on brassicas, on diseases, tomatoes, alliums, and we are still developing new ones on that, just general information of the state of the art. And this was all about the collection, the knowledge collection. Now we are working, as I told you, on the benchmarking, because from all those techniques, we want to identify the most promising techniques for the end user. And then we will use these techniques or we test them in case studies throughout Europe. So the first step we did in the benchmarking was to ask the end users and the experts and the advisors, we call that the experts and the stakeholders, and how many information they already had on the smart techniques. Is it important for them or not? And this questionnaire was in 12 different languages, and we had 4 main categories. So we asked about their professional, their knowledge on the techniques, the behavioral and the technical information they had. And finally, 17 countries visit the survey website. So some of them were filled in online. The others one we phoned up, we called the people and filled in together. And finally, 322 participants completed, with the highest participation in Spain, followed by the UK and Slovenia. And this gives us a lot of information. So the A circle here is a stakeholder survey, including the end users, and the right, the B are the expert survey, so the researchers. And if you're looking what is the knowledge on the smart IPM technologies, then you can see that most of them are quite little informed and only a few are very well informed. But then we asked, is it important for you? What do you think about for the future? Then most of them say it's important or it's very important. So a very few say it's not important at all. And if we analyze the results more in detail, then we see for all the 4 subgroups we had, or groups, categories, application or monitoring, that most of the people say that they currently did not use any of the techniques at all. So it's quite innovative. Sometimes there are differences between the countries, but especially in the southern countries, it's not common to use drones or spray robots. Actually, we are doing the benchmarking exercise now, so all the partners are filling in these forms. So we are scoring the techniques and giving in the final score, and then we work with the SpiderRap chart. So this is done by our German partner GKI, who is responsible for the benchmarking, and we are finishing this report by the end of this month or beginning of March. And this brings me finally to the end of my talk. And the last year we will focus on the knowledge exchange. So we'll have a lot of open days in the different countries. Different partners are working out programs on training sessions, seminars, and we will finalize the project with the Smart Protect Innovation Rally, which is organized by Inagro. Here you can see my colleague Jonathan, who is informing farmers on smart traps, which we were testing out this summer. And I also want to finish with this slide, which shows the links of Smart Protect with other EU projects through Europe. So thank you for listening to my talk, and if there are any questions, I will be happy to answer them.
Many thanks, Sabine. I don't think there are any questions in the chat at the moment. Just to say, and I put it in the chat, if you're aware of any interesting smart tools that we haven't identified, then please let us know. Obviously, the focus is on IPM and in vegetables. But apart from that, yeah, really interested to hear about anything. Okay, so I'm now going to give a short presentation on current issue. There's strange noise. Right. I'm now going to give a presentation on current issues for pest and disease management in field vegetables in the UK. And really, I'm going to talk about issues rather than solutions, just to see whether there are amongst the audience any sort of common areas of interest that we might follow up later on. Before I go on, there's a question from Raina saying only in field crops or also in greenhouses. Yes, we're looking at both, Raina, field crops and greenhouses. And obviously we had a session on greenhouse crops like this last week, but Smart Protect covers all vegetable crops. Okay, so I'm going to try and share my slides. Okay, can you see them, Sabine?
Yes, we see it.
Great.
Great, yeah.
Okay. Okay, so what I forgot to say, but was quite, I think, obvious when you all registered, was that this meeting has been put together on behalf of UVRIN, the IOBC working group on integrated protection in field vegetables, and the Smart Protect project. So, as I say, I'm going to talk about current issues for pest and disease management. And I think the main reasons for the current pest and disease issues are, first of all, loss of pesticide approvals. So some of those have had a significant impact recently, and particularly in the last few years, the banning of the neonicotinoid seed treatments. A general lack of effective control methods for some pests and diseases, and some of these are actually long-term problems where you know, we and others have been working on solutions for quite a long time. And then there are new pests or pathogens. These might be actually new to the country, so invasive species, but quite often they're old pests or pathogens that become more damaging just because of a change in the management system. So the first pest I'd like to talk about that is certainly an issue at the moment is the bean seed fly, which is a complex of 2 species, and it's always been a bit difficult to control, but now because the seed treatments on the most susceptible crops are being lost, it's a big issue, and it's very hard to manage in other ways. And Becca, my PhD student, is going to talk later about her PhD project on bean seed fly. Another major issue at the moment for vegetables is virus in carrot, and that is a direct result of the loss of the neonicotinoid seed treatments. And in recent years, there was a bad outbreak year in 2020. There were yield reductions of up to 30% because of virus, so very significant. We have a project on it at the moment, and we're looking at which vectors are critical and how spray treatments should be timed. And then there are longer term questions about where the virus comes from and how it moves through the cropping system. So the role of wild hosts and overwintered crops, because in the UK we store our carrots in the winter in the field, so we overwinter the crops. And we've also got an interest in potentially looking at sources of host plant resistance. A new old pest is the swede midge, and that was flagged up in 2019 by growers. They thought it was becoming an increasing problem in brassica crops. And the main impact is blindness of crops, distortion of the growing point. It's a native species to Europe. It's always been around, but it is a new pest in North America. It arrived there about 20 years ago and it spread enormously. So they've been doing quite a lot of work on it, which is very useful. So for the UK, what we've had for the last 2 years is a collaborative monitoring project with the aim of increasing our understanding of the problem. And we've now got some reasonably comprehensive sets of information from pheromone traps, which can inform what we might do in future. Then we move on to flea beetles, and there are, if you like, two types of flea beetle that are important in our vegetable crops. One are the members of the family Phyllotreata, and there are about 8 species that feed on brassicaceous crops, and generally they tend to be considered together. And they're particularly a problem in crops that are going to be sold as, I guess, as sort of salad crops. So things like pak choy, and you can see very damaged pak choy from one of our field trials last year. And then the other problem is cabbage stem flea beetle, and that has become a big problem because of the loss of neonicotinoid seed treatments used to manage it on oilseed rape. And I think that's had knock-on effects on vegetable brassicas, and it particularly causes problems on sown brassica crops because there isn't really any way of controlling it at the moment. And then just to move on to some diseases, then the different forms of fusarium have caused problems in a range of vegetable crops, and our focus here has been particularly on onion and on lettuce, and it causes root rots and wilts, and there are races that break plant resistance, and these can evolve pretty rapidly, and it can also survive for many years in the soil. And our particular interest at the moment is in Fusarium oxysporum lactucae, so the fusarium that attacks lettuce. And there are a number of races of Fusarium oxysporum lactucae, FOL. So there's race 1, which is quite widespread and causes disease in outdoor lettuce, and that occurs in sort of warmer locations. And then what we're particularly concerned with is race 4, which has emerged very recently in Northern Europe, and at the moment is affecting protected lettuce only. And then the final pathogen that I'm going to talk about is Pythium violi and related species, and this causes what we call carrot cavity spot in the UK, and this is one of the long-running problems together with fusarium in onion that colleagues have been working on for a number of years. So recently, colleagues have been working on the development of diagnostic tools, on the epidemiology of Pythium, looking at the effect of environmental factors and other things, and then also looking at biofumigation for control of cavity spot disease. So in terms of solutions, then there have been in the UK recently a couple of programmes that have had quite a broad focus. And the first one has been the Scepter Plus programme, which has been managed by our levy-funded organisation, the AHDB, the Agriculture and Horticulture Development Board. And the aim of the Scepter Plus programme has been to evaluate both synthetic pesticides and biopesticides for the key targets that the growers have identified. And this programme has covered the whole of horticultural crops, including ornamentals. And it's mainly consisted of efficacy testing and then support for approvals, working all the time with the companies. And this programme finishes in March this year, so we've only got a month to go. As some of you may know, the growers have decided not to fund horticulture anymore through the AHDB, and so the sort of future of programs like this is a bit uncertain at the moment. But these are the targets.
Rosemary, you are muted now. Just the last sentence from this slide. Yeah.
I must have hit something.
Okay, yeah.
Okay. So these are the targets that we've looked at in the Scepter Plus programme. So these are, I thought you'd just be interested to see what the growers have identified as priorities. So lettuce root aphid, bean seed fly, asparagus beetle, onion thrips, several species of aphid, flea beetles and cabbage whitefly. And then diseases, septoria, fusarium, downy mildew, neck rot, cavity spot, leek rust, pythium and rhizoctonia. So you could say that these are the problems that are of most concern to growers in the UK at the moment. Oops. And the other programme that has been running for a few years is called the Vegetable Genetic Improvement Network. And this is linked to the UK Vegetable Gene Bank, which is based here at Warwick University. And this is funded by our government, so the Department for the Environment, Food and Rural Affairs. And the idea of this programme is to identify useful traits from genetic resources. And then if we identify a useful trait, to work with seed companies so that they can breed those traits into commercial varieties. And these are our targets. Again, I thought you might be quite interested. So clubroot in brassicas, again, fusarium in lettuce, aphids in lettuce, cabbage root fly in brassica, diamond bat moth, aphids in carrot, and then fusarium in onion. So obviously new pesticides, biopesticides and host plant resistance are two good and complementary approaches, but we do need further work on complete IPM strategies. And one such approach, which colleagues have been investigating, is combining partial host plant resistance with biological control. And the project on the right-hand side, led by Dave Chandler at Warwick, has been looking at such an approach to manage the aphid pests of brassica crops. But aside from new pesticides, biopesticides and host plant resistance, then physical and cultural approaches may also be very useful. So I'm going to stop there. So I'll just say thank you to everybody who's helped with the presentation. And then just to say, please email me if you'd like to exchange ideas about particular pests or diseases of vegetable crops. So thank you. Okay. Right, so I see there's a question from Marita about what about, I can't even say it, Theophilopsis on carrots in the UK. Is it a problem? I'm not aware that it is, Marita, but I will ask I'm an entomologist, I'll ask my colleague, John Clarkson, who's a plant pathologist, and if it is a problem, I'll let you know by email.
It's a fungi. Sometimes we have it in Belgium.
Ah.
Yeah.
Okay. Yeah. So does anybody else want to comment about it?
I just know that this fungi is a big problem in Switzerland. And so therefore, I saw also that some people from Agroscope are joining today. And so they might know it under this, or it has another name as well. And so they might know it. So if you have a question about that, I think that you can contact the people from Agroscope.
Excellent. Okay, thank you. And I say, if anybody's got any more information about it, please put it in the chat. So we're now going on to a presentation by Ute, and she's going to talk about the IOBC working group on integrated protection in field vegetables. So over to you. You're muted, Ute.
Oh, now it should work. So,
yeah, fine. And we can see your slides, so that's great.
Great. Okay, so it was not about cats, but it's about the IOBC VPRS working group integrated protection in field vegetables. This group is, at the moment, I'm the convener of this group, together with the co-convener Hilfred Huiting from the Netherlands. He's working in Wageningen. So this group started in the 1980s with the main focus on integrated plant protection methods, which were new in these days. And the main focus is or was in this time the biology of insect pests, the monitoring and forecasting tools, mainly in cabbage crops, carrots, and leek and onion. So it was mostly that this group wanted to encourage, on one side, the applied research to work on integrated pest management techniques and tools, and on the other side, to implement these techniques also in the commercial production to produce high quality vegetable crops. So this group has the focus on collaborating and discussing the different approaches, and therefore to develop together or in these discussions IPM tools which can be used in the commercial production. And therefore this group combines applied research, this group exchanges with scientists, advisors, and also with vegetable producers. And in our working group meetings, we have mostly parts in the program when we have the different talks from the different groups. But we have also excursions to field sites, to vegetable producers, in the different countries to see also what's going on in the fields. In 2017, we met in Switzerland and we visited field vegetable producers and also greenhouse production site. And also in our last meeting in 2019 in the UK, we visited also different farmers. And in this, at that meeting, we discussed mainly IPM in field vegetables with approaches in pest monitoring, decision support systems, also landscape management, agroecology. And the IOBC working group offers the platform and the possibility to connect and to collaborate either with projects which are financed or just financed within the groups, so that there's an exchange and a collaboration to work together and to have a common output. And especially in the last years, we had a project, or it was a European project, the Fly IPM project, where several groups which are regularly attending the working group meetings, which were working together to improve integrated protection measurements to control vegetable flies. Here we have some impressions from our last meeting in the UK, from the different sites we were visiting. And mostly we have the challenges and the developments which are discussed in our groups. Our focus also on climatic change, how different pests and pathogens might be influenced, whether they are whether they have a better condition or which are like disappearing in the field. We have the importance of integrated pest management, like Rosemary already told in her talk before, that changes in pest management influence also the presence of different pests and pathogens in the field. And here we want to discuss and to exchange the knowledge and how different pest management strategies are used in the different countries. We have also not just climatic change and changing pests and pathogens in the field. We have also different field vegetable crops. We have new cultivars. We have new vegetable crops which are produced in the countries, and therefore we have also new challenges what pests and pathogens might occur in the field. And so we want to exchange about our research activities, about the knowledge we have, about the experience, and to achieve national and European goals. We need to exchange so that we have the experience from the other countries, from other research groups, and we can try this also in other in our countries. For the future activities, we planned a meeting last year, but due to COVID, we cancelled it. And so this year we want to meet in September, from the 20th September to the 23rd September in France, in Rennes. And the local organization is by Anne-Marie Cortesero and Vincent Faloya. And again, we want to improve our knowledge about the different cultivation practices in the different countries, also by having an excursion to production sites and to see what's going on and what's new in the different countries, because there's still the necessity of applied and scientific discussion to strengthen the development of integrated and alternative plant protection methods, to support sustainable farming practices, to support the production of vegetables, to support the income of the producers, and also to meet governmental and social expectations to secure field vegetable production in our countries, and therefore also for our nutrition. So that was it about the IUPC WPRS working group. And I'm looking forward to our next meeting. And also if someone has questions, I'm there to answer them. Thank you.
Excellent. Thank you very much. So I guess just to say that we can keep you informed through your email addresses, if you like, about the details of the meeting when they become available. Rainer, you've got your virtual hand up. Did you have a question?
Oh, I'm fine. That was on purpose. No, thank you. Okay,
brilliant. Right. Okay, so it looks like there aren't any questions at the moment. So thank you again, Ute. So now we're going to New Zealand. Brilliant. And so Graham Walker is going to talk about why IPM programs in outdoor vegetables have been successful and failed in New Zealand. So over to you, Graham.
Thank you. Am I sharing?
Not yet.
Content from camera, presenter mode.
You all right?
Can you hear me? So if I click the arrow up, that's the one that takes me to sharing. And it's given me all these options: share, presenter mode, content only. Sorry, browse my PowerPoint live.
I guess presenter mode might be all right. I don't know.
Yeah, presenter mode.
Add background. What sort of screen?
Otherwise, I can try to share your presentation, Jack. I think it's work, yeah. Oh,
okay,
no.
sorry. Yes.
You see your screen? Yeah, it says,
Oh,
yeah.
got it now. Yes.
Got it now?
Yeah.
Sorry, there were too many instructions.
Don't worry. Don't worry.
That. Okay, so you've got it there?
But you need to put it in presentation mode if you can. So slideshow. Yes, brilliant. Well done.
Okay, sorry about that.
No problem.
It's getting very late. Thanks very much for the invite. I didn't really, I heard from others, Meta Nielsen, that there was this interesting web workshop, and I do IPM and outdoor vegetables, so it's a bit of a surprise to get the invite to give a talk, but just a very quick intro. I'm an entomologist, been working on biological control of insect pests and outdoor vegetables for over 40 years, realizing that they were being nuked by broad spectrum insecticides. So then we moved about 35 years ago to IPM programs. But I've also, so that's in outdoor vegetables in New Zealand, but I've also spent nearly 10 years in the last 7 years doing IPM and safe vegetables in developing countries in the Pacific and Southeast Asia. But so I'll focus on New Zealand, and you're hearing me okay? And I'll move to the second slide.
Yes, all looking good.
All
Thank
going?
you.
Okay, right, yeah. We're off. So I'm really going to focus on biological control. I mean, I've spent 25 years looking at insecticide resistance and diamondback moth and helicoverpa and aphids and thrips and whitefly. But I just think the IPM is, I call it CC, compatible controls. And the most compatible, I think IPM became really practical and workable when we had effective biocontrol agents, and then along came the selective insecticides. So things like the BTs, and more recently the diamides, which really fit nicely into IPM. But I'm going to talk mainly about biocontrol agents because I think it's very hard to do IPM if you do not have effective biocontrol agents, and that's my focus of my talk. Two types, those introduced. Now, New Zealand has a really long history of introducing biocontrol agents. That's classical biological control. But the last 20 years, we've realized that we have these incredible resident biocontrol agents. Many of them are natives. We didn't before they were being nuked by broad spectrum insecticides. So it's a whole new ball game, and talk a little bit about those, the status of our IPM programs, and we've had a couple of recent failures in outdoor processing tomatoes and struggling with sweet corn pests. And I want to just give a summary of why I think that's happening. So just a very quick intro, because I realise I'm talking mainly to the other side of the world. But New Zealand, so I am in Auckland, and really we've had an endless beautiful summer for the last couple of months. But from Auckland north in the New Zealand map, which I hope you can see, we have no frost. It's nearly subtropical. And we can basically grow outdoor vegetables all year round, whereas in the south it's more like the UK. So our IPM research, in outdoor vegetables, we tried to do our scientific trials in large plot trials. We had very large research stations where we could have large areas, and the more we left crops untreated, the more we learnt. So mainly working on last 20 years, potatoes, but cabbage, cauliflower and broccoli and diamondback moth and white butterfly is what I've done for 30 odd years. outdoor head lettuce about 10 years. But on the East Coast, we have these important summer processing crops, processing tomatoes and sweet corn, and of course our warm and now hot summers where the pest outbreaks arrive. So just quick look at some of so I want to talk about effective biocontrol agents, because if they're not effective, they're almost useless. An effective biocontrol agent is very smart. So, for example, a long time ago, we introduced Cotesia rivercris on armyworm Thymus separata. And there's actually no IPM program for maize anymore, because we don't need one, because we get 99% parasitism and the population crashes. So no insecticides now get sprayed on maize crops in New Zealand because of effective biocontrol agents. About 15 years ago, down the bottom, we introduced Cotesia rubecula, which is in Europe. Before we didn't have it. And we now get, it's not unusual to have cohorts of 100% parasitism, so you get no big fat green white butterflies. Another very important, probably the most important one, which is quite world unique, is Cotesia kazakh, which we imported from Kazakhstan about 30 years ago. And it attacks and kills small larvae of Helicoverpa amygdala before they grow large and start going into the fruit. So very successful. And we also introduced Microplitis, which attacks those that are larger and escape that biocontrol agent. Those were introduced, and they're mainly larval parasitoids because they are specialists. New Zealand's quite strict on introducing biocontrol agents. They need to be specific. But what we found when we started not spraying our outdoor vegetable crops were these amazing predators, particularly these two here, a native hoverfly and a native lacewing. This native hoverfly builds up during spring, and in early summer is completely dominant. It eats everything on all leafy vegetables. There's nothing else left except big fat hoverfly larvae. It's incredible. Various spider species. Can you still hear me okay?
Yes, yeah, fine.
Oh, yeah, okay, sorry. I just so quiet here talking to myself in this room. Spiders are really important. It's only when I started working in Southeast Asia, and then I came back and saw that ground spiders and other spider species are incredibly important. They can protect all your transplants, seedlings, small plants. And then when the plants get bigger, you have these foliage-dwelling predators. And we have two of them. So the hoverfly comes in, and it also eats out this, probably the most important one down the bottom, the native brown lacewing, which is Micromis tasmanii. It's also in Tasmania. They claim it, but we claim it as well. A little brown lacewing. This predator is around all year. And it's sitting, residing, living on vegetables, waiting for anything to turn up, and it eats them. So it's really dominant, and it's not only an aphid eater, it eats all sorts of things. So very important biocontrol agents. So the status, a quick summary of the status of our IPM programs, and our biggest success has probably been in vegetable brassicas and outdoor processing tomatoes until things went awry. In IPM vegetable brassicas, we have very high levels of parasitism and predation of our key pests, which are diamondback moth and white butterfly. As I said, introduced Cotesia rubecula, really good control. Our IPM program, which includes a diamondback moth resistance rotation strategy for insecticides, 80% uptake by industry and a 60% reduction in insecticides. We even eradicated Pieris brassicae from New Zealand about 4 years ago. That is the first record of an eradication of a butterfly pest anywhere in the world. So good collaboration between industries and growers. IPM for outdoor processing tomatoes. World famous, not only in New Zealand, but probably the most successful introduction of parasitoid against Helicoverpa armigera. I'm sure you all know Helicoverpa armigera. So many common names, old world bollworm and tomato fruit worm, corn earworm. We introduced Catesia kazak. We get up to 90% parasitism. It kills the larvae while they're small and only eating the leaves before they grow bigger and bore into fruit and corn. So we had a 90% reduction in insecticides, 100% uptake by growers in industry, but it failed. But I'll talk about that in a second. Also, IPM for potatoes doing very well. Head lettuce and sweet corn, bit of a problem. So just quickly going through the important biocontrol agents in some of the crops. So in vegetable brassicas, I gave a quick summary, but for Plutella xylostella diamondback moth, we introduced the larval parasitoid, Diadegma and Diadromus, the pupal parasitoid. But we found that we had a very virulent strain of a naturally occurring fungus, which can take out diamondback moth in human conditions. And then, as I said, we find that these hoverfly larvae and spiders will take out pests when the plants are small. So some of the biocontrol agent interactions in brassicas. But this is the one I just want to spend a minute on because this was published. There's lots and lots of papers and an incredibly successful story until the disaster happened. So I just want to spend half a minute on this. So Catesia IPM in outdoor processing tomatoes, we introduced Catesia kazak. It kills the larvae before they grow large. It's all based around scouting crops and action thresholds and all that. But if you look at the graph, we introduced the parasitoid, and over 20 years we're looking at this, what happened. Mid-80s introduced the parasitism up to about 80%. Sprays on a tomato crop down from 9 down to basically 0. We got a little bit of resistance in the early 90s. But then if you also look at the changes in insecticide use, we moved from the broad spectrums to the BTs and the spinosads. And as I said, we basically got insecticide use on outdoor tomatoes down to 0, which was quite amazing, really. Now, here's a sad story. That IPM program, which was basically world leading, is dead. And this is because of tomato potato psyllid arrived. It's spreading around the world. It's now in Australia. It's the psyllid that vectors a bacterial disease that destroys tomato and potato crops. Unfortunately, on tomatoes, because of the trichomes, predators do not like tomato leaves. So we have no effective biocontrol agents for the psyllid. And although that's a potato crop on the right, that's what the tomato crops look like now if you don't spray them. So that new incursion destroyed that IPM program. Actually, to go to potatoes, we have a pretty sound IPM program. Again, the classical biocontrol, introducing Catesia sabantinus on potato moth. But when the psyllid arrived and we couldn't grow potatoes in New Zealand for a couple of years, just completely destroyed. Tomatoes and potatoes were completely stuffed because of that psyllid. We, at our research station, continued to sample unsprayed potatoes for 3 years. And this is where we found these predators. In winter, the lacewings, and then spiders and these hoverflies. Same pest, except this time, unlike in tomatoes where it just destroys, it looks like a virus and destroys the tomatoes, in potatoes, it spreads zebra chip, and that just completely destroys your potato crop. But this is my last significant slide, just 30 seconds on this is our winter, July, right through to autumn. And here's an early potato crop. and we counted everything on untreated potatoes every week for 3 years. And what we found is that this brown lacewing is the dominant insect on potatoes from winter, when we can grow all year round from in Auckland and a bit north, right through till early summer. Then this hoverfly comes in and completely dominates. So in December, in unsprayed potatoes, we can have 200 predators per plant. That's eggs and larvae. Quite unbelievable. Unfortunately, in summer and late summer, the psyllid goes berserk, and we're still working on controlling that. But basically now, no insecticides go on potatoes until summer, which is pretty amazing. And one other failure, we spent probably 20 years trying to do IPM in outdoor sweet corn. It's mainly a summer crop. But we've really failed. They're still using calendar spraying. And most of the problem is, it took a lot of work, that wonderful parasitoid of Helicoverpa, Catesia kazak, which kills 90% of small larvae in tomatoes. Well, you can walk across, you can jump a fence and go into a sweet corn crop, and it's destroyed by Helicoverpa because this parasitic wasp will not move into corn. And because of that, we have no IPM program in corn. Plus we have Nazara, which is also, we don't have effective biocontrol agents. So in summary, why has IPM in outdoor vegetable succeeded or failed? Well, the success is due to compatible controls, and that is effective, not biocontrol agents, effective biocontrol agents along with selective insecticides. Plus, of course, all that background research. We've done 30 years in brassicas and 10 years in potatoes. So background research, proving monitoring systems. We've got actual action thresholds, industry buy-in, and the hardest thing to do is implementing your IPM program. But our failures to finish were due to new pest incursions, the behaviour and crop preferences of the biocontrol agents. Some crops are effective, some they are not. And so our failures, talking as a biocontrol entomologist, is actually lack of effective biocontrol agents. Thank you. I hope I haven't gone over too much.
No, you're fine. Absolutely fascinating. Yeah, really.
So world famous in New Zealand.
Yeah.
Now, we've done a lot of publishing, so there is a lot of published stuff, particularly in the tomatoes, potatoes, and vegetable brassicas.
Yeah, no.
Thank you.
Thank you. Really, really encouraging, and discouraging at the same time. So I just had a question, because I always thought New Zealand was pretty careful about letting new organisms in. So what's the process for introducing biological control agents?
Well, I started as a technician in the biological control group with my guru, Peter Cameron, and it used to be specialists only. So basically only parasitic wasps that were proven to be specific to a target pest and nothing else. But there has been a change. So we now have Environmental Protection Agency, EPA, and you can actually now balance the pluses and the minuses. So if you can show, as long as you're not going to threaten a native species, so that it might, you like a native psyllid, for example, if you want to bring in we brought in a biocontrol agent against the exotic psyllid, you have to prove that you're not going to be other than a little bit of harm. So you're allowed, so the benefits have to heavily outweigh the negatives. But I would say it's a little bit easier. So for example, we have just introduced Tamarixia triose from Mexico, the US, to try and control that tomato psyllid, and yet it will attack native psyllids. But we've shown that the location of the any risk is really minimal, partly because they are non-preferred hosts, but also they dwell in Southland or the highlands in the South Island, so they're not going to come across this biocontrol agent, which is sitting in potato crops in Northland in the north. So sorry, I should have said. So there's a there's a balancing that can, so you can justify bringing in something that isn't an absolute specialist, whereas probably when we were doing exactly that, they had to be specialists.
Interesting. And there's another.
Although, sorry, I should say that in the 30s and 40s and 50s, we brought in I didn't, otherwise I wouldn't be here. But we brought in coccinellids and lacewings and predators. But that's because we have a long history. But from the 70s, we really only introduced specialists. But it's just opened up a bit now. So we've just let in something that isn't a specialist. But so a lot of background, probably 5 years of quarantine and host status testing to see host status, non-preferred hosts. So a heck of a lot of quarantine testing of insects on insects and things like that.
Excellent. Yeah, really interesting. So there's another question here from Gary Bradbury. Do you now have any new insecticides for Helicoverpa which are more selective, less harmful to parasitoids than, say, indoxacarb?
Oh, absolutely. Same. I now work mostly in Cambodia, Vietnam, Tonga, and Fiji, but New Zealand, and I am promoting the diamides. So we talk about the decades of the neonicotinoids, were probably 2000, 2010. And I think the last 10 years has been the decade of the diamides. So cyantraniliprole and chlorantraniliprole. I actually did confidential, but I'm allowed to say the results. Assays for DuPont and Syngenta to test their diamide products in New Zealand against the natural enemies, and showed that they were harmless to adult parasitic wasps, and very little impact with residues. So we did dry residues, wet residues, direct contact on predators, parasitoids, and all that. So short answer is that so we say use BT and other biopesticides early, but BTs are no good on large caterpillars. I mean, we even had we did GM. We had BT potatoes and BT cabbage, and we used the toxins, and we looked at them all in quarantine. The problem with BTs is they're not very good on big caterpillars. So this is where, so you can use BTs early in your IPM program, and then you use your diamides, but you have to be very careful because already in Southeast Asia, the diamides are nearly finished because of resistance. So the other thing you've got to do is rotate. So we still use indoxacarb, we still use the spinosins and the diamides and the BTs, and we have a rotation strategy. And that's actually the key to the IPM program is you start with your you always start and use your selectors as much as possible. Start with the BTs, then a diamide, but you only use maximum of what they say 2 or 3 times. And if you sometimes in Southeast Asia where it's 40 degrees, their pest pressure is so high that sometimes they have to spray quite a lot. So then we say after 3 diamides, you must switch to a spinosin, and then you switch to indoxacarb. So rotation strategies are it, plus using your selectives early, BTs and diamides.
Excellent. And then there's a final question, which is from Anne-Sophie. Are nuclear polyhedrovirus viruses, NPV viruses, commonly used against Heliothis armigera in New Zealand?
No, but I have a hot off the press. we're quite jealous of in Australia, the NPV virus for Helicoverpa has been around for a very long time. Because it was a virus, for some reason, so the answer is no. But we are now working with Endomat and Biocontrol at looking at a new virus for Plutella. So we're just starting to do the regulatory work. So we've never imported a virus, and I should have said, in an IPM program, because they don't use those viruses in Southeast Asia either. So that would be one of my bio controls that I would use in an IPM program for Helicoverpa if we had it. So of course, you use all the tools that you have, and we don't have that tool. Great,
thank you. Right, I think we better move on now. So thank you very much indeed, Graham. I hope you can stay with us. Or perhaps you want to go to bed. But
no, very interesting. But
thank you very much. So just to say, before we move on to the next presentation, there's a general question for everybody in the chat about information on IPM in sweet potatoes and then thrips in beans. So if anybody has any information, please type that into the chat. So now we're going to go on to Becca from the University of Warwick, and she's going to talk about IPM on the bean seed fly. Over to you, Becca.
Thank you. Can you see that, or would you prefer it a different way?
No, that's absolutely fine. Well done.
Okay, great. Okay, thank you. Hi, nice to meet you all. I'm Becca. I'm Rosemary's PhD student at the University of Warwick, and the title of my PhD is The Biology and the Integrated Management of the Bean Seed Fly. So today I'm just going to introduce the bean seed fly and tell you what it is and why it's a problem. I'm going to focus on two objectives of the PhD to conserve time. So I'm going to be looking at the overwintering biology of the bean seed fly and predicting its emergence, and then I will conclude. So bean seed fly is a complex of two species in the family Anthemiidae, Delia platura and Delia florilega. In the UK, it's a problem, well, all over it's a problem because when it's a larvae, it feeds on the seed and stem of emerging and germinating crops. It's a generalist, but in the UK we have problems on legumes and alliums. And it's a problem because there are a lack of effective insecticides to control for it, so a more sustainable integrated pest management strategy is required. So that is the aim of the PhD, is to contribute towards an integrated pest management strategy to reduce crop and economic losses by the bean seed fly. I've got 4 objectives, but today I'm focusing on objective 1, which is to investigate the impact of temperature on the development and to look at forecasting the spring emergence of bean seed fly. So what have I found so far about the overwintering biology of the bean seed fly? So we already know that as a fly, it goes through 4 distinct life phases, and as an insect, its development is entirely driven by environmental drivers such as temperature and day length. There are 2 other really common agricultural pests, the cabbage root fly and the onion fly, which are related to the bean seed fly, and we know those go into diapause over winter. If you look in the middle, we've got a life cycle. For example, if we were to rear bean seed fly at 20 degrees, it would take 2 days for eggs to hatch as larvae, 14 days for them to pupate, and then 15 days to undergo that complete metamorphosis into an adult. And so what do we already know about their pattern of activity? So this graph here is showing bean seed fly activity in blue and cabbage root fly activity in orange from the beginning of 2019 to the end of 2021. And we can see over winter that bean seed fly activity is reduced and cabbage root fly activity, well, there isn't anything. And we already know that cabbage root fly go into diapause. So are bean seed fly going into diapause, or is something else happening? And then this graph also shows us that bean seed fly emerge in large numbers in spring, and this coincides with growers in the UK planting their crops, which then is a problem. So can we predict that spring emergence? So under the overwintering biology objective, I'm just going to present the results of 2 questions I asked. So do bee and seed fly enter diapause, and if so, how long do they spend in diapause? And all of this information is going to help understand how to predict their spring emergence. So to answer these questions, I cultured some eggs. For experiment 1, I placed the eggs outside in environmental conditions for 280 degrees because that's how long we estimate it takes them to develop into pupae outside. The pupae were collected and then emergence was recorded at 20 degrees. For experiment 2, I reared them in the lab at 10 degrees for an 8-hour photo period. And then for 62 days, they were reared and pupae were collected. And then they were placed at 0 degrees for different durations. And then emergence was recorded at 20 degrees. From the data I asked, did the adult flies emerge in 15 days at 20 degrees? And asked the question, did they emerge in 15 days at 20 degrees? Because we know it takes them when they're developing normally to emerge in 15 days at 20 degrees. So then we assumed they were not in diapause. And if they didn't emerge in this time, we were assuming that they were in some form of arrested development. So in answer to the first question, the results are currently suggesting that yes, they do go into diapause, but only towards the end of late September, which is really interesting. So this graph here is showing batches of eggs that were placed out at different time points from the beginning of September to October. And those that were placed at the end of September, less than 50% of those emerged by day 15 at 20 degrees, suggesting a large proportion were in diapause, compared to those that were placed out before and after. And we did this experiment on wild bee and seed fly, well, a similar one in 2020 to 2021, and we found similar information. Those that were laid towards the end of September, and this was at Warwick Crop Centre, they showed less than 50% emergence by day 15 at 20 degrees, suggesting that a large proportion of those were in diapause. However, those laid after potentially didn't go into diapause. In answer to the second question, our current results are suggesting that they go into diapause between 0 and 50 days. So this graph is showing bee and seed fly that were reared at 10 degrees to initiate diapause and then placed at 0 degrees for different periods of time. And this was just to mimic different lengths of winter. And we found a significant difference between those that were placed at 0 degrees for no days and those that were placed at 0 degrees for 50 to 150 days, suggesting that diapause for those that do go into diapause lasts between 0 and 50 days. So current conclusions under this objective is that a small proportion enter diapause. Their diapause is relatively short, but a proportion do not enter diapause. And again, this is all to aid predictions of their spring emergence. And the implications for forecasting of all of these results so far is that there is potentially some bee and seed fly that are not going into diapause. So we need to understand how we sort of monitor or forecast their emergence, because a lot of forecasting models assume the insect is going into diapause. And they focus on accumulating day degrees, so we need to work out when is the best time to accumulate those from. So then going on to the model. So just to recap, we want to predict the spring emergence of bee and seed fly, that's shown in the graph in the bottom left there. And many models focus on the accumulation of day degrees. And for those who don't know what day degrees are, it's the accumulation of temperature over time. So my research question was, can the accumulation of day degrees be used to predict the spring emergence of bee and seed fly? And why is it important to do this? Well, this can help growers aid decision making. So maybe they can sow at a time that is avoiding when bee and seed fly are active. The model used 5 years of bee and seed fly activity data from water traps. Each year we predicted the start and end of the emerging generation from bee and seed fly counts, and then day degrees were calculated using soil temperature, base temperature of 3.9 degrees, and were accumulated from the 1st of January. And our results say, yes, there's a statistically significant relationship between the accumulation of day degrees and the proportion of flies to emerge in the spring generation of bean seed fly. So I fitted a lot of different sort of models to the data. We found that the viable type 2 3-parameter model is a good fit as per the residual standard error, which just tells you the goodness of fit to a data set. And what we can do is get an equation from this data to predict different proportions of emergence. So currently we can conclude that accumulated day degrees can be used to predict the spring emergence of bean seed fly, but we really need to now start thinking about how the information we've gained from those overwintering experiments can be used to aid predicting the spring emergence more. And we also need to validate the model. So we need to compare it to different years and in different locations in the UK. And again, this is all to aid farmers make decisions. So conclusions so far overall. So under the overwintering experiments, they're suggesting there's a diapausing phenotype and a non-diapausing phenotype of bean seed fly. Under the modelling experiment or the models, there's a significant relationship between the accumulation of day degrees and the proportion of flies in the spring generation to emerge. However, we just need to test the efficiency of the model and focus on encompassing, maybe we need to make a different model for those that are not going into diapause. So yeah, many thanks for listening. Yeah, really happy to take questions. And yeah, thank you to the organisers of this meeting and Rosemary, my supervisor.
Excellent. Many thanks, Becca. So I don't think there are any bean seed fly questions at the moment. If anybody is doing any work on bean seed fly or is finding it a big problem, then please get in touch, because we'd be interested to talk to you. There are a couple more sort of general questions in the chat for people to probably respond to in the chat, although if we've time, we can have a discussion at the end. So Anna's asking if there are pheromone traps for Nazara or other traps to monitor it. And then there is a question as well about whether anybody has any suggestions about natural barriers or trap crops for IPM on thrips. So something for you to think about. Quentin, did you have your hand up, or is that?
Yes. Yes. Hello. I just wanted to react on what you said, just for short. So we start working on the bean seed fly this year. So we're going to start with that. We want to also try to develop rudimentary modules for the SWOT model. Maybe we will be able to integrate some modeling modules in the SWOT model. We will have field work, and I'm very happy to get the opportunity, or to have the opportunity to get in touch with Becca, because I have several questions on how get this fly, how get the rearing or breeding of these flies, and would be very happy if we can get in touch maybe outside of this.
I'll put my email in the chat for you.
Yeah, thank you very much. Yeah.
Thank you.
Excellent. Right, that's a good connection. Graham's got a question as well.
Yes, it's actually more a comment to Becky. You don't know how lucky you are, because we have Delia in Southeast Asia. Mainly, they call it onion fly. I'm not sure of the species, but there are a couple of Delia species, and they're absolute monsters. And of course, there's no cold winters. They just keep going, and you know, it's 30 in winter and it's 40 in summer. So I'm just saying, yes, I know of problems, and thank goodness for cold winters.
Yeah, it's quite interesting. Yeah.
Yes. Comment Nazara. I don't think we've done a lot of work on Nazara. We're waiting for the brown marmorated stink bug to arrive, so we're practicing on Nazara. But I don't think there's a pheromone, but there's an aggregation pheromone for Nosara, but no, it's a very hard one. Those pentatoma very hard to control.
Great, thank you. Okay. Right, so they're also in the chat, which is good. This is really good because this is what I was hoping would happen. So also something from Anne-Sophie saying that bean seed fly is a big problem in France and maybe need to collaborate, and then from Sarah in Sweden as well, saying that. So probably what we can do is arrange a sort of mini meeting with a few of us interested in bean seed fly at a later date. So that's great. Excellent. Right, okay, so thank you very much, Becca. So now we go on to our next presentation, which is from Neringa, and is integrated and alternative plant protection in Lithuania. So over to you, Neringa. Yeah, I can see your slide.
Hope you see my slides, so I will start. Are they moving? Okay. So yes. I am from Lithuanian Research Center for Agriculture and Forestry, and the main center research areas are agriculture, horticulture, and forest plants. We are located in several locations of Lithuania, so we cover all the area and we can get a lot of data. I am from Institute of Horticulture, Laboratory of Plant Protection, and our research topics are biology, ecology, epidemiology, diversity of pathogen and pest species, biological studies on relatively new pathogen and pest species in relation to changing climate, integrated plant protection, efficiency of forecasting models for pests and diseases in horticultural plants, applications of biological and alternative plant protection products in horticulture, and also efficiency trials of new plant protection products according to GAP standards. Integrated plant protection in Lithuania basically started from forecasting models, and with forecasting models we are working from 2007. There was several models only. Now the Lithuanian Advisory Service bought more forecasting stations, and the net of forecasting stations are quite covering all the area, and the farmers get the information in electronic service of information consultation and training platform. For integrated plant platform, there they can find information about dynamics of disease and pest and weed spreading, integrated plant protection, catalogs of plant protection products, diseases, pests, weeds. There are some photos of pests, and also they can get the remote education. Also, we are preparing IPM guidelines for farmers. It's been prepared for several crops. Now we are going to prepare for strawberry, cucumber, and tomato. And I think these guidelines quite helpful for farmers because they get all the information about one crop in one place. So it's quite good for them. We use Imetos forecasting stations from Austria, PESU. So these stations has a lot of sensors. In our institution, in my lab, we are working more on apple scab, strawberry green mold, onion botrytis, and carrot alternarium forecasting models. And our research showed that using forecasting model, the spraying's application dates were more precise compared with conventional and traditional. So the sprayings are better according to forecasting model. We spray for disease for a special time, so it's optimal for disease. Also in apple fruits and also for vegetables, we are working on plant protection system, which we call national systems. So the same active ingredients has to be not used more than 2 times per vegetation season, and the pre-harvest interval is 1.5 times longer compared with indicated on the label. And also you cannot use the products labeled as very toxic and toxic. So the sprays according to the systems gives quite good results, and the apples were quite good. The apples cap was completely covered, so it's quite good results, and we think that the system is quite good for farmers. Also, we have to look for new pathogens, so the epidemiology, distribution, and identification is the main goal. So we are looking for new pathogens, monitoring them, and seeing some recommendation for farmers. Also, we are working on essential oils and plant extracts. We think that plant protection should be focused on natural products, and the whole world is going on that. So we did some research on plant extracts and essential oils, and essential oils quite are promising. However, there is not a lot of substances registered, but it's the long way to go. So we think that will be in future. So our research showed that Thymus vulgaris essential oil completely inhibited Coleotrochum acutatum, Botrytis cinerea, Fusarium oxysporum, Alternaria dowsi, and other pathogens. So it's quite promising product, but we have to make it we have to make the product. Also, we did some experiments fields in strawberry with some extracts, clove extract as mono with water mixture, and clove laurel extracts mixture. So the results are quite promising also. It's only the third year, but damaged berries amount are less compared with other treatments. So it's quite promising, and we think that we have to do more experiments to get better results. Also, plant extract and essential oils could be used for seed treatment. Because seeds are contaminated with various pathogens, so seed treatment I think it's one of the future trends. And our results showed that Alternaria on current seeds we were quite suppressed by Thyme essential oils. So the Thyme essential oils is quite good for seed treatments, but in future we have to do more experiments. So the other trend is intercropping plants. Because there is some ecological farms who wants to grow a lot of vegetables in one farm. So the phytoncid plants, I think, is quite good for them. And we get the result that phytoncid plant did not affect the mineral composition or the main macronutrients like nitrogen, potassium, phosphorus, magnesium, calcium of cabbages. And our results suggest that marigold, French marigold, common Thyme, common Saja, phytoncid plants can be grown together with white cabbages to improve phytosanitary conditions. So the future application is with drones. It's not regulated yet in European Union, but it's going. So I think it's future trend for precise application. For application in precise time and place. So we hope that we can use less water for sprayings and suppress our diseases also good. So the visible light in agriculture and horticulture is undeniable because it's induced photosynthesis, which is needed for plant growth and development. The light has effect also on fungi sporulation, stimulation, or repression, suppression. So LED light, besides plant growth, has also an option to select specific wavelength for targeted pathogen control. Light quantity and quality parameters can determine pathogen and bioecological characteristics on pathogenesis process. So we did some research on Botrytis cinerea and Coleotrochum acutatum, and we got the results that different photoperiod and different wavelength differently acts fungus on this. Also, we found some good trends which can be suppressed, and we are doing future next our experiments are on lettuce and strawberries in greenhouse to suppress Botrytis cinerea in greenhouses with light. And I invite you to the 3rd International Conference on Scientific Actualities and Innovation in Horticulture in Vilnius. And so, thank you.
Thank you very much, Neringa. What a lot of information. Amazing. Thank you. So, so I've got a question about the intercrops, the companion planting. What is the spacing of them?
It's normal spacing. You grow 1 row cabbages, 1 row phytoncid plant, 1 row cabbages. It's like normal, but it's one beside the other. So it's same system, same growing system, only 1 row cabbages, another some marigold or some flower crop.
Okay, so the growers don't mind that half the field is taken up by the companion.
For ecological, no. Ecological, they want to grow everything ecologically to get more. They are a little bit small farms, so. But I think for future we have to think something because pathogens and pests are coming, and some phytoncid plant we can use for aphids. They can attract them and not go to our crop. So.
Yeah. So do you know which pests they work? Which pests and diseases the companion plants actually reduce?
We looked for Alternaria. But our results still have to be worked. But it's a little bit suppressed, not quite big, but some amount of glucosinolates, which suppress diseases. We are higher with some marigold and tagetes. So we think that we should grow them together. Maybe when glucosinolates are higher amounts, so they are more susceptible to diseases.
Interesting. Thank you. Yes. So there's a comment from Marita, which means she says it means you have 50% less yield from the same area, but I think what you mean is the cabbages are the same size if they.
Yeah. If we use the less space for crops, yes, so the yield will be less. But for ecological, you get the price bigger. The price is higher, and you can get.
Yeah, great. Okay, and then Gary's I think made a comment more than a question, just to say he agrees with your point about plant extracts, essential oil. And the lack of them registered in Europe. He's saying that in countries like Mexico and Colombia, there are at least 12 different plant extracts registered for use, which makes the IPM approach.
In Europe, I don't think that we have product. We have some components. I found only several components of essential oils, which are registered. More substances are on Bacillus subtilis or Trichoderma, or some viruses like pepinomosaic virus. So. But I think it will grow because pesticides are not working now because we are going to some European regulation to lower pesticides. So we have to find some alternative.
Yeah, no, very interesting. And then Marita's just asked what price is now for eco cabbages in Lithuania. It's hard to compare between countries. So, but what?
Now I cannot say because it depends on year, on each season. So
yeah, no, difficult one.
Yeah.
Okay, so I think we've covered all the questions. So brilliant. Thank you very much indeed for your presentation. And we'll go on now to the next presentation, which is Fabio Dorn. And talking about biocontrol in Spanish avocado orchards. Over to you.
Thank you very much.
Okay, we see a presentation.
Perfect. Yeah, just let me know if the slides may be full.
No, we can see your slide.
Okay.
Yeah. Okay.
So hello, I'm Fabio. Welcome to my talk. And currently I'm a master's student, and today I'm going to present you my bachelor thesis. Oops. I'm quickly going to put the pointer. So today I'm going to present my bachelor thesis in which I tried to understand how global warming can influence biological pest control in orchids of this plant here, the avocado crop. And therefore I studied if temperature modulates intergrid predation via behavioral changes. So global warming is influencing, for example, via drought, the worldwide productivity in agriculture, and also plant protection methods like biological pest control are affected. Why? Because in biocontrol, pests are controlled by its natural enemies, and both of them are mostly arthropods, to be known ecothermic. The natural enemies can change their efficiency to control these pests at higher temperatures. In nature, pests, natural enemies, and also other organisms are embedded in complex food webs which are affected by these temperatures. So a good tool to study these complex food webs are community modules. And the community module focuses only on the interaction of interest and simplify the complex food web, for example, to predation or to competition. And intergrid predation is the common community model which combines competition and predation between two predators. Therefore, these two predators share the same prey, and one of them feeds on the other one, and also the other way around. So commonly, the intergrid predator are adults of one species that prey on the juvenile stage of the other species, the intergrid prey. In literature, it is discussed if intergrid predation can influence and even interfere biocontrol. So the first question we wanted to answer was how temperature influences these trophic interactions. Therefore, experiments were conducted at the Agricultural Institute La Mayora, located in Andalusia in southeastern Spain. And this area is quite unique for Europe because it's the only subtropical region in whole continental Europe. And logically, therefore, it's also known to be the biggest European producer of exotic fruits like mango and avocado. And in the following, I will focus on avocado crops because I used the avocado mite community as a study module. And this community consists of a herbivorous mite and two predatory mites, of which one is augmentatively introduced to the orchids and one is naturally occurring there. So Oligonicus persei, the so-called persea mite, is a main pest of avocado. And that's visible through the damages on the leaf through necrotic spots. And those damages can be even reason for strong defoliation. And the white spots you see here on the right side are silicon nests built by these persea mites. And these silicon nests protect the persea mite against their natural enemies because those are not able to enter the nest, and also against harsh abiotic conditions, so they create their own microclimate inside that nest, and therefore they're protected against high temperatures, and also those nests can interfere chemical control because it's difficult to apply synthetic pesticides there. So the predatory mites of the persea mites potentially consist in intergrid predation. And Neoceros californicus is a specialist predator, which is able to enter the nest and also prey on the persea mite inside its nest, while the omnivore Eoceros stipulatus is not able to do that, so it's not able to enter the nest, and also not, and therefore only able to prey on the persea mite outside its nest. And besides the persea mite, stipulatus is also feeding on pollen. So first, like I said, it was necessary to find out how temperature affects these trophic interactions. Inma, one of my Spanish supervisors, investigated during her PhD how temperature affects the strength of these trophic interactions via fitness traits. The results of Inma's thesis were the background for my studies. So she studied with two different community blocks. how these interaction changes from mild conditions at 22 degrees to hot conditions at 30 degrees. And for both blocks, she predicted an intergrid predation. And to simplify these interactions, she predicted for the first block an intergrid predation consisting of the omnivore adults as intergrid predator and specialist juveniles as the intergrid prey. And the second block was inverted, so the specialist was the intergrid predator. For neither of the two community blocks, she found actually intergrid predation. The realized interactions in the first block with the omnivores intergrid predator at mild conditions was a trophic chain with 4 levels. And on top, you could find the omnivores intergrid predator. This trophic chain changed at hot conditions to a competition between the predators for the herbivore. So in the second block with the specialist intergrid predator, for either mild conditions and hot condition, a trophic chain with 3 levels was found. At mild conditions, the intergrid predator, at mild conditions, the intergrid prey was preying on the herbivore, and the intergrid predator was not interacting with any of the other species. And at hot conditions, the predatory mites switched roles, so the IG predator was not involved in the trophic chain, but now the specialist intergrid predator was preying on the herbivore. So those were the results of INMA, and based on those, I wanted to determine in my bachelor thesis how temperature-dependent behavior can be as well a mechanism for the discovered trophic changes. Therefore, I studied in behavioral bioassays looking like that. 13 different variables, but to keep it simple, I will only show you in the following those 5. And all the variables have been statistically analyzed with generalized linear models or generalized linear mixed models. And those behavioral bioassays consisted of a leaf disk arena that we picked up, that was stamped out of a fresh avocado leaf from the orchid. And we tried to have one nest from the herbivore inside of this arena, and then we added predatory mites from the lab in different combinations to those arenas. Now we come to my results. So for the first block, with the omnivorous intergrid predator, we had again this trophic change, changing to a competition at hot conditions. And I first focused on the behavior of the herbivore in both temperature conditions. And we hypothesized that the herbivore is more available to the predator and more preyed by them at hot conditions. The results showed that the herbivore tend to spend more time outside the nest at hot conditions. And here on the right, you can see visually that, here on the right, you can see visually that at mild conditions, the intergrid predator preys the intergrid prey, as here. But at hot conditions, the intergrid predator moved inside of the nest and So here on the right, we visualized, so I was confused with the slide, sorry. Here on the right, we visualized the trophic effects of these behavior changes, and we concluded that at hot conditions, the herbivore is more available to its predator outside the nest. And this could be then also a reason for the higher predation by the omnivorous predator at hot conditions. Secondly, I studied the behavior for the specialist intergrid prey to answer if it would be less available and therefore less preyed by the intergrid predator at hot conditions, and the intergrid prey tend to spend more time inside the nest at hot conditions. And here on the right, you can see visually that at mild conditions, the IG predator preys the IG prey more. But at hot conditions, the intergrid prey moved inside the nest and is therefore protected against the intergrid predator. So with the results for the omnivorous predator, I could support that it attacked less on the intergrid prey at hot than at mild conditions. The intergrid predator preferred to prey on the herbivore over the intergrid prey at hot conditions. And again, to make it more visual, you can see here that at mild conditions, it preyed on the intergrid prey, but at hot conditions, the intergrid predator preyed on the herbivore. And now we switch to the second block with the intergrid predator as the specialist. And in this block, we saw at mild conditions that only the intergrid prey was preying on the herbivore, and at hot conditions, only the intergrid predator was preying on the herbivore. I observed the behavior of the intergrid prey first to see if it's interfering with the foraging activity of the intergrid prey, of the intergrid predator on the Persea mite. And I used the number of encounters between the intergrid prey and the predator as an indicator for the interference. So the intergrid prey encountered the intergrid predator significantly less at hot conditions. Reason for that, we assumed, was probably the significantly lower number of escaping attempts and at mild conditions. So at hot conditions, the intergrid prey escaped more from the intergrid predator. And here again, the visualization. So at mild conditions, the intergrid predator, the intergrid prey was interfering with the intergrid predator, which was therefore not preying as much on the herbivore, and other individuals of the intergrid prey could also therefore consume more the herbivore. At hot conditions, the tables turns and the herbivore is preyed by the intergrid predator because the intergrid prey was escaping to colder microhabitats and therefore also not interfering anymore with the intergrid predator. So generally, we could assume that the behavioral strategies of the juvenile stages of those natural enemies were decisive for the observed trophic changes. And in the first block, the specialist intergrid prey triggered the herbivore to higher exposure to predation at hot conditions. And in the second block, the omnivorous intergrid prey stopped to interfere the specialist intergrid predator, the more efficient biocontrol agent, because it's a specialist, which then could resume to prey the herbivore at hot conditions. So here. And even if it seems now that hot conditions are actually beneficial for biocontrol, following population dynamics experiments in the field and also the interaction strengths experiments by my supervisor Inma showed that biocontrol was in this case hampered by an increased temperature. So after all these intergrid predations and hot and mild conditions, what is the take-home message for you now? In a nutshell, the success of biocontrol in future climate will depend on phenotypic plasticity of the biocontrol agents in both their fitness and behavior traits, and not only in fitness traits as most current literature has focused on so far. And if you want to read more on this topic, please have a look on our manuscript once we published it. Yeah, now I would like to mention and thank all the people that helped and supervised me during my thesis. And I would also like to thank you for your attention and the possibility to present here. So I'm open for questions and please go ahead.
Many thanks, Fabio. That's really interesting topic. So it shows how complicated the situation is. So how, if you were managing a biological control program, then how would you use that information?
To maybe adapt in future lines of the biocontrol agents which are more adapted to the to those temperatures. So that's also what they are right now doing in Spain, that they are also kind of doing experimental evolution to breed for new lines at different temperature conditions.
Okay, so selecting within a species rather than using a different species.
Or you could also go for different species. That's also possible.
Yeah, yeah. No, really interesting. Right, I don't think there are any more questions at the moment. I'm just going to suggest that we have a 5-minute break to fill up coffee cups and things. So if you can come back at 10:30, and then we'll have the last two presentations and see how much time we've got for a bit of discussion. Okay, see you in 5 minutes.
Okay, good.
Right. Hello again, and I've now got a cup of tea, so that's good. Go on to our next presentation, which is Anne-Sophie, and I can see you there already, which is great. And Anne-Sophie is going to talk about growers and industry in France funding for applied research on vegetables. So over to you.
Okay. Hello.
Hello.
Can you see my screen?
Seeing your screen, you just need to go into presentation mode, I think. Yeah, brilliant. Looking good.
Hello, everybody. I'm Anne-Sophie Coissy. I'm the technical manager of Unilet, which is a growers and processors organization in France, an association. And firstly, I present my organization briefly with some quick picture. Around France, we grow our veggies in 3 main areas where the facilities are set either for canning or for freezing. We grow 15 crops, but 3/4 of the crops we grow, 3/4 of the areas are French beans and peas, and they are grown on 1/3 of the acreage of total vegetable surfaces in France. 6% is grown under organic agriculture conditions, and we represent 4,000 growers, and they are gathered into 18 producers organizations. So our end missions at Unilet are dedicated to the specific goals of the processed vegetables. And for that, we are located, our research centers are located in the 3 main regions, close to the growing and processing areas. And we conduct experimentation in field experiments, research, and also demonstration activities. Our missions are to support the sustainable development of the sector, helping to reduce the use of PPPs, to stimulate the upmarket of the production with labels or organic agriculture label, and also to look after new crop protection solutions. We are funded by the members, and 65% of our financial input is from self-financing, and our team is constituted of 7 agronomists and 9 technicians, and we also deliver technical information to professionals via reviews or internets. I wanted to share with you my experience of why the veggies are specific when they are grown for industry, and what it draws as hurdles or levels for IPM. Firstly, we have a long crop succession, and we alternate our vegetables with main crops, and that means that some techniques are not very easy, but we can work on them to increase the IPM in veggies. Cover crops, tillage conditions are not yet well used by producers, but we can work on this progression. But as we have a long crop succession, we can easily manage soil-borne pests and weeds, and we can also give advice for field selection before sowing. As our crops are sown and they are high densities with narrow rows, and they must address the industrial planning, it's very difficult to increase mechanical weeding, and it's also difficult as there is a planning of sowing and harvesting for industry. It's very difficult to avoid some pests at some times when they occur at some times, but it's also a chance because as we are organized in producer organizations, there is a possibility of collective, not risk management, but risk insurance. Then we are open field, we are full season grown crops, we get climatic variations, and we have a lot of pests for that, various pests, and it's difficult in open field to use management, to use beneficials because they don't go where we want them to go to fight the pests. But we can easily manage water and fertilizers. We can you can work on pest surveys and decision tools. We can test the genetic varieties which are more resistant or tolerant to the diseases. And of course, we can work on biocontrol, either in soil or spraying. And also, as the standards, quality standards of the industry are very high, because the crops are mechanically harvested, we can't use some levels of IPM, such as varieties or plant associations, and we must work with the industry to sort visually the defects on operation lines. I wanted to do a focus on how many trials we drove for 10 or 12 years at UNILET, only on the aspect of biological control and biopesticides. We conducted over 600 trials, and we tested around 100 specialties, out of which 60% are effectively marketed. So this represents 60 actives, and we tested it on 11 vegetable crops, but mainly, as usual, on beans and peas, because they are our main crops. There are all kinds of substances that we tested, natural ingredients from plants or animal or mineral sources, microorganisms such as bacteria, fungi, viruses, and microorganisms such as nematodes or trichograms. Main trials have been conducted on beans, and they met our main issues, which are gray and white mold, caterpillars, seed flies, or now more recently soil-borne fungi. Every currently authorized solutions have been evaluated. Our first a lot of work has been done on contents, which is Coniotherium minitans, or Bacillus or Trichoderma. And against caterpillars, we have tested the NPV virus, Bacillus thuringiensis. Commonly, 2 biological solutions are used in the sector. Upon 5 are really authorized. The biofungicide contents against white mold, and insecticides with BTs in organic farming only. And the 3 others, despite they are authorized, they have not demonstrated their interest or only in organic agriculture. In peas, the solutions are tested against downy mildew or aphids. And we have identified several solutions interesting, but there is no authorization yet to use them in the fields. These solutions are the phosphonate against mildew and the fatty acids preparations against aphids. And for these 2 targets, the satisfying control has been demonstrated for many years, but the limit is that the marketing authorizations have not still been given. And we still have only emergency authorizations to use pyrethrin and then fatty acids against aphids. And obviously, the main biocontrol product used on peas is sulfur against powdery mildew. Our 3rd crop of interest is spinach. And on this crop, 2 main issues have been addressed: the downy mildew and damping off. And very few solutions are allowed, and there are no prospects currently. Against mildew, 7 products have been tested, but are still not conclusive. And against soil fungi, 5 specialties have been tested, and only 1 is authorized and is not very efficient in the fields. So to date, no biocontrol agent has been proved to be interesting on spinach specific pests, and we don't have market authorizations yet granted. Anyway, some solutions are used in conventional farming and have been plainly adopted. And number 1 for us in France is Coniotherium minitans. The product is named Contents, which is widely used to control white mold. And we estimate that it is commonly used on around 10,000 hectares in France on vegetables. The second use is sulfur against powdery mildew, but it's very far behind Contents. It's only we estimate the area only to 4,500 hectares. And then the other ones are very underrated. We can evocate the ferric phosphate against slags. In conclusion, we have relatively modest results compared to the investment that the sector made to try and test biocontrol pesticides, because many products do not demonstrate their interest in the field. but anyway, if they do, the marketing authorizations take too long to emerge, and there is globally a lack of efficient solutions. There are some research projects in progress, and we also need to learn how to use the solutions when they arrive, because they are very different to be used from synthetic products, and we must take care of not disappointed growers by letting semi-good solutions arrive in the fields. So I thank you, and I hope my presentation will give you an occasion to exchange with us on your experience. Thank you.
Excellent. Many thanks, Anne-Sophie. That covered quite a lot of ground as well. First of all, there was a question from Rainer. Do you have no thrips problems in French beans?
Not in beans, no thrips problem. We have thrips problem in peas. It's not a main problem, but we are outdoors, so no, not on beans.
Interesting. And then another question about pests. Do you have any solutions for bruchus on beans?
I don't know what is bruchus.
Beetles, aren't they? Bruchid beetles?
Oh yes, not on green beans. There are only problems on field beans, but it's a rather a small crop for us. But it's a major problem for Fabé, but not on green beans.
And another pest question: do you monitor Helicoverpa armigera in France?
Yes, a lot. It's a major problem in the southern zone. So we monitor it, and we try to find solutions. If you want to collaborate, no problem. and they are coming upwards from the south to the north year after year.
So are they migrating then?
Yeah. No, yeah. They migrate each year, north and southwards, but also they progress northward year after year.
Yeah. Oh, I see.
They overwinter in France.
Okay, so they're overwinter and then they just move. Yeah. And then I had a question which Gary answered. My question was, is it more difficult to implement IPM in crops that are grown for processing rather than for the fresh market?
There are some differences, and no, not globally more difficult, but the problems are different because of the rotations and the crop successions, and also the size of the fields. And usually for industry, we have fields from 5 to 50 hectares, and some techniques are not easy to conduct in such big fields.
Yeah, thank you. And then another question: do you do direct seeding on French beans or conventional tillage? So are you doing no-till or no-till?
It's increasing. It's not common, but some growers make experiments or are convinced and try it. But there are problems of weeds anyway.
Yeah. Excellent. Right. Thank you very much indeed. That was very interesting. Just to say, there's a bit of a dialogue going in the chat about Nizara, but I'm sure people are sort of watching that. So now we'll go on to our our last presentation, which is from Sebastian. You can see Sebastian, which is about increasing the efficacy of natural enemies in field vegetable production by combining banker plant systems and tailored flower strips. So over to you, Sebastian.
Sorry.
You got me instead of your presentation.
Okay. Here we are.
Yep.
Can you see the slides?
See the slides? So you just need presentation mode.
Yeah.
Fabulous.
Thank you.
Great. Thanks a lot.
Thank you. Yeah, good day, everybody. I just want to introduce to you a new project that we recently launched. We did that with 2 partners. So one partner is Katz Biotech. It's a biocontrol agent producing company, and an environmental trust from Lower Saxony and Northern Germany, Stiftung Kulturlandpflege. So in this new project, we try to increase the efficacy of biological control in the field by combining 2 biological control strategies. The one strategy is banker plant systems, and the other one is tailored flower strips. So you probably are all familiar with flower strips. They provide basically nectar and pollen, and also shelter to natural enemies. So they promote functional biodiversity. These flowering strips may also promote pests, certain pests that also make use of nectar and pollen, so floral resources. So that's a special thing on tailored flower strips. So they aim to give nectar and pollen and shelter, so promote specific natural enemies, but minimize the use for pest insects. So for instance, lepidopteran pests also on the adults feed on nectar. The other biological control strategy that we use is a banker plant system. So what is a banker plant system? It is composed of the actual banker plants and alternative hosts of prey and the actual natural enemy. It can be one target natural enemy or more than one. So normally the banker plants are pre-infested before planted. So you plant them together with the alternative hosts of prey and the natural enemies on it. In the beginning of the season, when the pest is not on the crop yet, and the aim is to develop an open rearing in the close environment to the crop. And as soon as the actual target pest arrives on the crop, the natural enemies are already in the field and are ready to attack. So they should migrate to the crop then and reduce the pest population at a very early stage. And therefore, reduce also insecticide applications and damage and increase the yield. So what are the actual possible benefits of a combined use of target flower strips and banker plant systems? So the first is very obvious. So actually with a combined system, you provide all that natural enemies need, so shelter, nectar and pollen from the flowering strips, and also alternative hosts of prey for reproduction of the natural enemies. That makes this system quite independent from actual pest population, because the hosts of prey are always there on the banker plants, and also independent from the flower abundance in the local environment, because the flowering plants are already there as well. Furthermore, it's also independent from the occurring of the natural enemy populations. So it might be that the natural enemy population did not get through the winter well, or for instance, there are bad climate conditions in spring. So, and often it's often the case, or the normal case, that first the pest arrives and the natural enemy arrives later with a delay. So, but with a combined system, we are independent from the natural enemy population, because we release the enemies on the banker plants. Furthermore, the system is adjustable to specific requirements or situations during the season. So you always can adjust the banker plant system, so compose the banker plant system targeted to a specific target pest and a target crop. And also during the season, for instance, if you monitor and if you see that there is a lack of natural enemies in the cropping system, you can always reintroduce new banker plants and therefore new natural enemies. Often you have natural enemies in the system that require different resources at different life stages. So for instance, hoverflies, lacewings, or parasitoids. So the adults need pollen and nectar, so floral resources, and the immature stages need prey or host. And you provide everything with a combined system. And as I said, so it's quite specific to the target crop, and not only to one target pest like the banker plants alone, but to a pest complex of a target crop. And hopefully it makes it all more efficient. Banker plant systems are already quite established in greenhouses, or for instance, as we see here, cereals as banker plants with cereal aphids on it and parasitoids against aphid pests in greenhouses. It's a quite closed system without very much influences from the outside. There are optimal climate conditions for the natural enemies, so that works pretty well already. However, in the field, it's a very different situation. So you have many different factors that influence the system. So changing weather conditions, other natural enemies that might cause antagonistic interactions with your natural enemies that you introduce. However, during the past years, the research on using banker plant systems in the field has been increasing. So we also established a system in cabbage in some years ago in another project. There we came up with a banker plant system. So that was actually pumpkin plants as banker plants with the alternative host Trialorodes vaporiorum, so the greenhouse whitefly. These do not cause damage on actual target crops, so cabbage, so we can use them. And the main natural enemy of the cabbage whitefly, Alaerodes bulletella, that is Encarsia tricolor. So that was the system: pumpkin, Trialorodes, and Encarsia tricolor. And these are the results. So during one season, the banker plants produced more than 1,700 Encarsia tricolor parasitoids per square meter all over the season. Yeah, this led to more than 50% increase of parasitism on cabbage, on the cabbage whitefly, and yeah, the Banker plants also increased the abundance of hoverfly larvae on cabbage by more than 60%. Yeah, and all this led to a reduction of an cabbage whitefly population. So in another project that was done during the last years, a colleague worked on developing a tailored flower strip against cabbage pests. So today it's commercially available as the trade name Hannover Mix. So it's composed of 9 flowering plant species that you see on the right. Yeah, so he had an increase of different predatory groups and species, especially syrphids and coccinellids, but also increased the abundance of different parasitoids, especially aphid parasitoids and Lepidopteran egg parasitoids. And that led to a reduction of these pests, so aphids and Lepidopterans, by 30–40%, and also increased the yield in the end. So we use these 2 systems in the current new project. We see already that the effects of these 2 different strategies are complementary. So the Banker plants rather promoted, in the first instance, Encarsia tricolor. So it's in the first instance a biological control strategy against the cabbage whitefly, and this tailored flower strip, Hannover Mix, it's predominantly against aphids and Lepidopterans. So yeah, so we use this study system cabbage because it has also a very complex pest complex population. So it has the pyrus, butterflies, Clutella, and the aphids, and yeah, also the cabbage whitefly and cabbage fly as well. So the new project is funded by the DBU, so the German German Environmental Trust. So one working package works on improving, optimizing the actual Banker plant system, so the production of it, and hopefully will come up with one or more commercial products in the end. So for instance, the Banker plant system itself, so the species needs to be optimized because for shipping and so on, the pumpkin plants are quite big. So for producing the Banker plants, you need about 5 weeks to inoculate them with the alternative host and the natural enemies. And after 5 weeks, the pumpkin plants are pretty big. So we are looking for another plant to use, more compact growing plant. Then the alternative host parasitoid ratio need to be optimized. So you need, you want to have as many parasitoids as possible that you release into the field, but you also need the alternative host population to, not to decrease. Then the parasitoid sex ratio is very important with the Encarsia tricolor. So because the reproduction system is quite special, so Encarsia tricolor is a facultative autoparasitoid or hyperparasitoid. That means that females, mated females, use the nymphs of the unparasitized nymphs of the cabbage whitefly as primary host, and from these mated females, let eggs develop females, and if the mother female is virgin, so not mated, lays the eggs on parasitized whitefly nymphs, and from these eggs, males develop. So if we release mated females into the field, at a certain point, there's the risk that you have a female biased population, and so the proportion of these females that are not mated increase, and these do not contribute to biological control. So we need to develop, and we want to develop, a release concept. So the solution will hopefully be that we additionally release at a certain point males into the system. So we have to monitor intensively, and as soon as we have a very female biased population of the parasitoid, we introduce more males, so the females get mated and contribute to biological control again. So basically stabilizing the parasitoid population by this. So we want to come up with different products here. So this is the normal banker plant system with a banker plant, alternative hosts, and males and females of the parasitoid on it, just to establish the parasitoid population in the beginning of the season. Then this stabilizing product with banker plants only with male parasitoids on it. That's might be introduced later in the season when the population is female biased. And then we might also have a product that are banker plants only with alternative hosts on it that could be used to increase the alternative host population in case the parasitoids lack hosts in the field. Another working package will will focus on field experiments. So the first aim is to develop a monitoring system, because monitoring is very important here. So we need to know when the population of parasitoids is female biased, for instance, and also we want to monitor the whole biodiversity, what's the effect on it. So we use different trapping system to do this. And then in the first year, we want to determine the type of interaction. So how do they actually interact? So the flowering strip and the banker plants. Is it really a additive interaction, or even synergistic, or it could also be that it's an antagonistic interaction. So for instance, there could be an inter-guild predation. So the predators that are promoted by the flower strip could have a negative effect on the parasitoids on the banker plants, for instance. So so we try different spatial arrangement of the banker plants and the flowering strips. So right next to each other, as you see on the right side, or separate from each other. And then we, in the end, we want to come on with a practical application. And in the second and the third year, we go on to vegetable farms and actually evaluate the combined system in the practice. Yeah. Thank you all for the attention. And yeah.
Excellent. Many thanks, Sebastian. So I think that stimulated a few questions. So I'll go up to the top of the list. Right. So first of all, from Jonathan, what were the absolute numbers? And I think that means the numbers of beneficials in the Hanover mixed trials.
The absolute numbers of beneficials I'm not quite sure. So which beneficials was that? Sorry.
Jonathan, do you want to follow up? Are you there? No?
I will send him a chat, Rosemary. He has also a demo this afternoon, and maybe somebody is calling him because of the storm. People are just.
Yeah, okay, we'll go on to the next question. Thanks, Sabine. Then Uti had a question about, can promotion of hoverflies cause problems for vegetable producers because more hoverfly larvae occur in harvested vegetables? And then Gary has actually responded further down and said yes. Would that be your view as well, Sebastian?
Yeah, of course. I think the consumers probably don't want any insects on the product. So if they see hoverfly larvae on their cabbage, it's probably so they cannot distinguish between beneficial insects and pests. So that's not wanted as well. Yeah.
Okay, and the next one was actually me, which you then partly went on to talk about, is how do you decide on the amount and spatial arrangement of banker plants and flower strips? Because I think often the experimental design is quite challenging for these sort of questions.
Yeah, so that's another thing we need to figure out as well. So the ratio between banker plants, flowering strip, and the crop, actually, yeah. Yeah, so that will be done in the second and third year on the farms. So I think generally these experimental designs are very difficult with flowering strips. So because you need actually a lot of distance between the plots. And yeah, so on your experimental fields at the university, there's not that much space. So I'm looking forward to go into the field, into the practice, to have more space to look on that.
Yeah, and yeah, I think it's one of the most interesting experimental questions, really. But yeah, then there's a comment from Graham about you need to be careful that reservoirs, banker plants don't lead to an increase in hyperparasitoids that attack your biological control agents.
Yeah, that's actually why we want to introduce the males during the season, because if we keep the population of mated females high, that will decrease the risk of hyperparasitism in it. Okay. So if you have a high ratio of unmated females, so what they will produce males and look for parasitized whitefly nymphs. That's why we would to release the males as well.
Yeah, no, I mean it's interesting your approach. I'm just that the chat's got quite complicated now.
Rainer has a comment as well.
So yeah, Graham also says, as Sebastian says, yes, challenging. For Phacelia and potatoes, we tested single bed strips of Phacelia at different distances at the edge and into the crop. Yeah, so there's a question from Gary. Could you provide your email address, please, Sebastian?
Yeah, of course.
Yeah, and then also there's a comment from Torsten Schöneberg, Agroscope. We're planning a similar approach in lettuce this year. Would be great to connect with you and discuss your experiences further. So again, he'd like your email address.
I will send the email address. Just contact me.
Yeah. So, and then I think there's a bit of a discussion going on about, does anybody have experience in monitoring egg parasitoids, which I think probably.
I've been figuring that out as well. So I was contacting different companies that produce egg parasitoids, Trichogramma, for instance. So they gave me some instructions to build traps to monitor Trichogramma in the field. So we will try to do it with sentinel cards, so sentinel eggs of Cytotroga, so the moth eggs, glued on a piece of paper, and try different trap systems there.
Sounds interesting. So Rainer, you've got your hand up. Do you want to say something?
Actually, the question is already solved, but it was a question that you had with hoverfly. So it depends a lot on the variety of cabbage that you grow. So if the hoverfly is on the produce, yes, it might cause problems. But if it's not on the produce, like for example with Brussels sprouts, it will not cause any problems.
That's true. Yeah.
Yes. Can I make a comment? Can you hear me?
Yeah.
We have farmers who complain that the hoverflies are too good, and when they eat, they leave yellow, orange, frass or whatever, and it actually is not good for. It's not so bad on broccoli, but not good on cabbage crops. But my comment was, of course, it's not an issue on crops like potatoes, where you're not harvesting potato leaves. So again, you've got the differences in crop types and issues between leafy and tuber crops, etc. And I did make a comment, egg parasitoids are absolutely useless in New Zealand, but Australia, particularly in Queensland, have done a huge amount of work. In fact, when their action thresholds for spraying for Helicoverpa are based on egg parasitism. So they've really done a lot of work that you could find in the literature. Thank you.
Okay, thanks.
Thank you. Right, so yeah, good discussion around all of that. So I think we've addressed all the questions for you, Sebastian. Thank you very much. That was, thought-provoking presentation. So thank you. So we've got just a few minutes left now. So I think it would be, so I think this has been great today, because there's been so much interaction and different topics, but relevant to a range of crops. So yeah, it's just how I hoped it would happen, and hopefully people can make connections about different issues. I guess we can also we can probably check whether everybody's happy for us to share everybody's emails, and then we could do that if that would be helpful. I guess the other question we have is, are you interested in carrying on with meetings like this again? And if so, when and how often and which topics? So again, I think we could probably do a simple questionnaire, couldn't we, Sabine, to do that?
We can do, yeah. I will ask Lynn to set us up. And we also can provide a YouTube link with all the presentations which were recorded today.
Yeah, and we're also saving the chat.
Yes,
because I think there was some quite interesting information, useful information in there. So we'll probably edit out the comments about not hearing people and not seeing slides and things, but we'll keep the information there. So I don't know, does anybody want to make any further comments before we close, either in the chat or put your hand up?
I would like to say something. I would like to thank you for all the presentations. It's quite a lot. I found it quite long. Perhaps it's good to do it some more, because I find the information quite good, but the problem is it's quite long. So perhaps we can do it more. I saw a chat from somebody who said next winter. Why next winter? Why not earlier? And we take, let's say, no more than 2 hours or something, because it's quite a lot for now.
Yeah.
And for